So this part of the Flow Cytometry course is a demonstration video on our usage of this package called ggplate. So this kind of forms this part of the course. So it's mainly just looking at how we can import data in CSV format and map that onto plates of different sizes. So what we want to be able to do is map our data onto these plates or micro plates and produce this kind of layout where we have maybe a legend denoting the values that we have here in colors, but also the numerical values entered in there and scaled according to the size, depending on the plate, and dimensions, and what the parameters of the actual plot surface area as well. So what we can do is make a plot with discrete values like the previous one, or with labels like this for particular treatments as well. You can see the colors could also have a metadata incorporated there as well. We can do plates of different sizes as well, from 3 to 12 to 24, 48 to 384. And you can see that the legend also can vary as well, depending on what precisely is of interest. What we're going to do with the other code is import existing data. So the lab itself will actually look at your data, but we'll import existing data so you can see the structure of this data and then use these functions like the function plate plot, which takes in this data, it takes in the position from the well data inside this object that we've imported. It takes in the values, which correspond to the value column, and takes in the labels as well. And it also takes in the plate size, which can change according to what we have. So let's switch over to R to look at this in more detail. So what we can do is use the install packages function to import ggplate as written out there. So if we run this line, that will import all the packages from there. There may be some dependencies that you require as well, but we can just install them there and then activate the library with the command on line 10 and just check the package version that we have as well, which is 0.01. So what we're going to do is import this data continuous 96 on line 13. So what we can do is look at the structure of that data to see more about it. So what it contains is a 96 by 2 table with 1 to 96 values, and then also the labels of the wells. So let's look at that in more detail. So these are the well entries. So it's a standard 96 well plate. You don't have to make these up yourself or type them in yourself. You can actually extract them from this vector if you don't have it yourself. And then the other column is the values with the capital V. So that's what we're plotting as well. So what we can do is the function, you can see there in line 19, we're going to follow that. So it's plate underscore plot, which is a package within a function within ggplate. We're saying we're going to look at that data. And we're going to say the position is the well, the values of interest are the value column we had there. So the names must match. The label, well, we had it in as the value there, but we can actually change that to the well if we wanted. And the plate size is important as well because we have different plate sizes. So we have to put that in too. So we have 96 here. So let's try that. So there we have our plates, and you see this time we have the labels on them instead of the values because I changed the label parameter to be different. So we can change that now to be value with a capital V. So now you can see the values. And now we can add on things onto there. So you can see before we had the labels equals well, that's something we can change between value and well, depending on what we want. What we can do too is scale the legend. So that's just adding onto our existing function, 96, and then we add on limits equals C, let's say -1, 5, and our final round bracket. You can see there now the legend has changed from -1 all the way to 5. The same color scheme, but you see the plate colors have changed too. So we could also tweak that, and you can see the plate colors change further as well. So many of the values are actually below 1, hence the grays. We have also data for the 384 well plate. So if we put that in there instead, we need to change the plate size to 384 so it matches. And we can try that. And you see that the plotting area is now bigger, so you might need to move this a bit just to make it more visible. But you can see part of there being shown. So in addition to this function, we can also specify the colors. So there's an example here of different colors we could use. So what we're going to do is put that in there as an additional component, and we might change the limits to be 0 to 4. So let's try that. So you can see that's now going from black to purple to red to orange. And if we change that and just delete the last one, so just going black to red to purple, where the orange was at 4, now you can see that the purple pinky color is now at 4. So it just defines the range we're going to. So you could go all the way from simply, say, purple to pink here like that, and we just range across a single gradient. So you can see that there. We could also just do it simply as black to white without the hash sign. And that's a more simple way of putting in a color scheme in there. And another way of developing functions, a color scheme is to add in rainbow with a particular number of colors that you want, say 12, and a final curly round bracket. And that will visualize it as a rainbow there. So you see a very Flovistic plot there. So there's lots of ways. And if we want to maybe make that easier to see, we would just turn that down to three colors maybe instead of 12. Oh, okay. It's still quite not the best coloring scheme there. What we can do as well is also consider partial data. So we might have 46 wells instead of 96. So if we do that and look at the structure of that data, it's basically the same. It's 48 by 2 table, and it just contains our values, where some are NAs, and then the wells again. So what we could do is, if we did this on the 384 plate, it kind of would look a bit empty, but that should plot there as well. If we take our previous plate, yeah, you can see it only takes a proportion, so that's maybe overkill. 96, let's try that. And you see there, it fills some of it. And then we also have 48 well plate. So let's see that there. So now we can see that. If we try to fit 96 into 48, what will happen there is that we miss some of our data because it's matching the well placements. So some of that data will be missing. So you just have to bear that in mind. So if we go back to the previous plot, you can see the 1.19 to 0.08 on the right-hand side. So 0.08 goes down to here. So it goes down to F8. We can also do 6 well plates. So that contains more simple data. You can see there just a few different drugs and a few wells. And that might be the metadata that's being used to treat a particular bundle of cells. So we can change that then to have a 6 well plate, and maybe we could add in a title like that. As you see there, we have a much larger text size, and we can change the title size as well. Title underscore size. Let's say 11. Let's see what it does there. It's kind of small. So you can increase that to 12. So you can see that's bigger. So you can see there's quite a lot of things we can tweak to modify our plot here. We can add more color as well to make it more easily interpretable. We can also turn off the legend. So we just turn show 11 to be false. So if you use that existing plot false and you see no legend is there. And we can also change the label size to be bigger or smaller depending on what we want. So if you use 4.5, it's kind of small for this size of plot. If you turn it to 8.5, it's kind of bigger. And then all other options are 24 well plate. So we have this data discrete 24. Let's use that with plate size of 24. We'll put our legend to be true. And here the items don't exist. So let's look at that data again. Condition. There we go. So now we have, instead of well and value, we have conditions. So we put a condition in here. And so what we need to do is consider the number of colors we have present. So what we can do is just modify this color scheme to be a sufficient number of colors, so 24 colors, and change the label maybe to be condition so that it corresponds. And then we have sufficient colors, and we can see that the label size now was gigantic. So let's turn off the label because that's not going to fit. So you can see there we have the color scheme and the corresponding legend there to indicate what we're looking at. We can also scale the sizes, so we can make that bigger or smaller. So let's try that. So if we try scale 0.55, you can see now the boxes are smaller. If we try 0.15, they're going to be really, really tiny. And if you try 1.5, they're going to be too large. So you can see there, it doesn't make sense. So setting back to maybe 1.05 is kind of the best we can do here. That's even too much. So 0.9, that's about the maximum we can do. And then the final example here then is the number of rows in the legend. So if we see this existing diagram where we scale it to 0.8 and add the number of rows with the legend, we have 24 values. So we might try 4 for legend row. You can see the area is squeezed. Let's turn that to be small. Then the legend is more organized. That's too small. So let's turn it to 8 and scale it to be 0.5. So you can see now, yeah, we've changed the legend. So we have maybe 6 per row in the legend. Sorry, the number 6 per column in effect. So you can see that we can toggle different plots. And the key thing is that we just need to read in our data at the start that our data should contain 2 items, a value, and then the well. That's all we need to use this function. So let's discuss it more when we have the actual class.